Hi, my name is Gloria Roberts. Today, we shall be discussing the topic enzymes. Enzymes. Objectives of lesson. As the lesson progresses, the learners should be able to 1. Define enzyme. 2. Mention at least 6 characteristics of an enzyme. 3. Give at least 3 classes of enzymes and 1 example each. 4. List and highlight how an enzyme works. And number 5. Mention the different digestive enzymes in the body and their functions. What is an enzyme? Enzymes are biological catalysts that are protein in nature and help to speed up chemical reactions without being used up. From this definition, we can say that enzymes are produced from protein, and at the end of the reactions, we get the enzymes back. Metabolic reactions in the body are controlled by enzymes. Did I hear you ask me why or how? That is simply because enzymes bring down substrates in the body. So the main function of enzymes in the body is to break down substrates. Biological catalyst is any organic substance that can increase the rate of the chemical reaction in the body. Therefore, organic or biological catalysts are found in both plants and animals and are usually needed in small quantity. Let's talk about the characteristics of enzymes. Number 1, enzymes are protein in nature. 2, they speed up or promote chemical reactions. Take note, enzymes don't start reactions. Number 3, they are organic catalysts. 4, they are soluble in nature. 5, enzymes work at specific temperature, usually ranging from 35 to 40 degrees Celsius. 6, they function at specific pH. 7, enzymes are specific in their actions, meaning a specific enzyme for a specific substrate. 8, they are unchanged at the end of the reactions. 9, they are involved in reversible reactions. And number 10, not the least though, enzymes can be inactivated by inhibitors. This means that enzymes can be prevented from functioning properly or functioning at all by the presence of substances called inhibitors. So let's discuss the classes or types of enzymes and their functions. Enzymes are classified based on 1. Where they function in organisms. 2. The reactions that they take part in. And so we are going to start by looking at enzymes classified based on where they function in organisms. Number 1 type here is called intercellular enzymes. These are categories of enzymes that work within or inside the cell. They are also known as endoenzymes. They are also known as what? Good, endoenzymes. Now looking at the image, the enzymes are found inside the cell. That is where they do their own function. Examples include ATP synthetase, DNA polymerase, and so on. Number 2, we have the extracellular enzymes. These are the enzymes that carry their own functions outside the cell. They are also known as exoenzymes. From the image, the enzymes are outside the cell, so they carry out their own function outside the cell. Examples include pepsin, maltase, sucrase, lipase, and so on. Now let's look at the classification of enzymes based on the reaction that they take part in. We have 4 different types we are looking at. The first one is called hydrolyzing enzymes. You can simply call them hydrolases. These enzymes work by adding water molecules to substances during reaction, and that reaction process is called hydrolysis. Or simply by removing water molecules from reacting substances by the process known as condensation. Note that all digestive enzymes are hydrolysis enzymes, and examples include amylase, lipase, protease, cellulase, and so on. Number 2, oxidation reduction enzymes. You can as well call them oxidoreductase. These involve the addition of oxygen and the removal of hydrogen during reactions as the case may be. The addition of oxygen is called oxidation, while removal of hydrogen is called reduction. Most respiratory enzymes are oxidoreductase. Examples include oxidase, dehydrogenase, catalase, and so on. Number 3, transferase enzymes. These enzymes transfer functional groups like the amino group, acidic groups in substances during reactions. Examples here include alanines and deaminase. Deaminase is an enzyme that is used in the process of deamination by the liver. Number 4, ligase enzymes. These enzymes help to join two molecules together. For example, we have molecule A and molecule B. Now they can be joined together by this kind of enzyme and they will become AB. Okay, common example here is DNA ligase. This is an enzyme that is used for joining the DNA of bacteria with the DNA of human in the production of insulin in biotechnology. So let's come to this mechanism of enzymes. How do enzymes work? To understand how enzymes work, we need to understand the following concepts. Number 1, activation energy. Activation energy is the energy needed to start a chemical reaction, meaning that for any reaction to take place, it must first attain activation energy. So no activation energy, no chemical reaction. However, when an enzyme is added to the reaction, the enzyme reduces the activation energy, thus speeding up the reaction. From the graph here, you can see the red curve is very high. Now that is a normal reaction that has attained activation energy. So it's going to take a longer time. Now the blue line represents a reaction involving enzyme. So the enzyme reduces the activation energy and so the reaction takes a shorter time. Number 2, active site. This is the space on an enzyme that is shaped to complement the shape of the substrate so that they can fit precisely. Yes, I believe you also got that. Active sites are located on the enzyme. Good. Now take note that active sites are also the points where the substrate binds with or fits precisely with that of the enzyme. From the image here, you can see the enzyme, you can see the substrate, and you can see that shape on the enzyme. That is called the active site. It complements the shape of the substrate so that they can fit together when added together. Number 3, we are looking at substrates. These are substances which the enzymes act upon to bring about a reaction. In other words, these are the substances that have been broken down by the enzymes in the body. Examples include starch, protein, fat and oil, maltose, sucrose, lactose, and so on. Number 4, products. Products are the result from the breakdown of substrates in the presence of an enzyme. So, when an enzyme breaks substrate, what are going to be formed are called products. Take note that the products don't usually look like the substrate. So from this image, you can see the enzyme, the substrate, they bind together at the active site, and what is formed, product, totally different from how the substrates look. Number 5, we have inhibitors. These are substances that can deactivate enzymes so that they do not function as expected. Earlier I said that inhibitors are substances that prevent enzymes from functioning. So how do they do this? They can either block the active sites of the enzymes so that the enzymes don't fit, the substrate don't fit with the enzymes, or they can change the shape of the enzyme so that the enzymes or the substrate cannot fuse together. Examples of inhibitors are mercury and cyanide. So let's look at the mechanism problem. We have 2 mechanisms we are going to be discussing. The first one is called the lock and key mechanism. This principle here is based on the fact that enzymes are specific in action. We said that a specific enzyme for a specific substrate. And so they function just like a padlock and its key. If you want to open a padlock and you don't have the right key, they cannot open, it cannot be opened. So they must complement one another. The shape of the key must complement the shape of the padlock so that by the time you turn, it opens easily. That is how this mechanism works. Now, so the enzyme's active site is shaped to complement that of the substrate. We have mentioned that earlier. So the two bind perfectly to bring about a chemical reaction. Now the enzymes are not used up at the end of the reaction. So from this image, you can see the enzyme, you can see the substrate, you can see the shape on the enzyme, which is called the active site. And when these two combine together, a reaction is set up. And at the end of the reaction, you are getting 2 different products, okay, that are totally different from how the substrate look. Something is common. Yeah, the enzyme is gotten back intact just the way it was. So it's not used up at all. The second mechanism is called induced fit mechanism. In induced fit hypothesis, the active site of the enzyme is not shaped to complement the shape of the substrate. However, the active site of the enzyme is flexible and can change its shape to a complementary one that can bind precisely with the substrate. Did you get that? Okay, let's use the image here. Now you can see the image of the enzyme, you can see the substrate. Now observe the enzyme very well. The active site of the enzyme is not shaped to complement that of the substrate, so there is no way they can fit together. However, in this mechanism, the enzyme can change its shape, and that is exactly what it's doing at the next stage. Okay, it changes its shape to that can complement the substrate, and the reaction is set up. Now at that time, the enzyme substrate complex is formed. As the reaction continues, enzyme product complex is formed, and at the end, the products are formed. Look at the product color difference, showing that they are totally different from how the substrate looks like. So the enzyme is gotten back at the end of the reaction. So let's talk about this. Note. Very important that you take note of these 3 points. Number 1, at higher temperature above the optimum temperature, enzymes can be denatured or destroyed. Optimum temperature is a specific temperature for an enzyme to function properly. Now, any temperature that is higher above that, the enzymes will be destroyed or denatured. So most enzymes function best at optimum temperature of 37 degrees Celsius. I didn't say all enzymes, I said most enzymes. Now, the second point here is that some enzymes work best in acidic medium, while others in alkaline, and a few others in neutral medium. Now, some enzymes like the pepsin work best, functions best in an acidic medium, such as that of the stomach. Now, alkaline medium, like the saliva, produces an alkaline medium for the ptyalin to function in the mouth. Now, outside this medium of these specific enzymes, they can become denatured. So outside their specific pH condition, they are denatured. And number 3 points, enzymes only work for a specific substrate at a time. A specific enzyme for a specific substrate. For example, amylase will only act on starch. Pepsin will only act on protein. Lactase will only act on lactose. Sucrase will only act on sucrose. So there are others like that, like ptyalin will only act on, we only act on starch, and so on to form maltose. So take note of all these. Questions can be set from here as well. Alright, let's move to the end. This is the student activity. We have 10 questions for you to test your mastery of this topic. All you need to do is get your pen, get your paper, and start answering. Okay, maybe you're not able to answer 1 or 2. Please don't worry. Go back to the video, listen to it, take notes if you have to, jot something down, and of course download if you have to, and at the end you'll be able to answer all of this and even more from this topic, enzymes. Okay, it's time for you to mark yourself. Our goal is always 10 over 10, or should I say 100%. But please, if you are new on this channel, subscribe to my channel, click the notification bell, like my video, drop your comment, and do any other thing you can do, or you can do to promote the video. You can share as well, download, do anything. Okay, at this moment I'll say thank you for joining me in this class. There is a classroom, and thank you for sharing your time with me. Thank you. Bye. Have a wonderful time.